ATP. And in so doing, it uses fuel, glucose if it has it, fatty acids or amino acids if it doesn't. And so it increases your metabolic rate, your use of oxygen, and so on. So it affects almost every cell in the body. But because of the metabolic discussion, we'll talk about it again at the end of the semester. Thyroid gland is located just below the voice box or the larynx. And it wraps around the trachea. So this is the cartilage of the voice box. It's called the thyroid cartilage, but it's not the thyroid gland. And it's typically H-shaped with two lobes and a elevated area here known as the pyramidalis in the middle. The isthmus is the cross piece. All right, so if we look at it, and then it wraps around from the back. So these would be a lobe, the isthmus, another lobe, we'll just have a brief discussion of the third endocrine organ, the parathyroid glands, which are found on the posterior, not inside the thyroid, but are adjacent to it, kind of shrink wrapped with it by connective tissue. Now, if you notice on the diagram, there's a high number of blood vessels associated with the thyroid gland. We're not going to learn those in this unit. But does that make sense? What do we associate endocrine glands and hormones with?
Blood.
Blood. So we have a lot of arteries bringing the amino acids in or the cholesterol, depending on the type of hormone produced, and obviously a lot of veins carrying those hormones away. So endocrine glands tend to be very vascular. I'm sure when you studied histology last semester, you looked at, this is one of the more common illustrations of simple cuboidal epithelia. It may look stratified where two sets are together, but each of these is a section through a sphere. So think of a basketball, not that big obviously. And if you section that sphere near the tip or the edge, you're going to get a small cross section. If you cut it in the middle, you're going to get a larger one. So that's why we see these different sizes. And so it has a wall of simple cuboidal epithelial cells. Some of those cells may not appear to be simple. When I teach histology, I don't like many of the illustrations that show simple squamous because they show simple squamous like that. That's really an inactive simple cuboidal. So you'll see simple cuboidal cells that look like this, and you'll see simple cuboidal cells that look like that. These are inactive. These are active. Just like an osteoblast versus an osteoprogenitor cell. Osteoblasts are synthesizing the protein collagens. It takes more cell organelles to do that, so the cell is bigger. Right? Now, unlike most of our other endocrine organs, the hormone produced by the thyroid gland is produced and not directly excreted into the blood. It's stored in this central area here in an inactive form. And then depending on TSH, TSH regulates both the thyroid stimulating hormone from the anterior pituitary. It will make more, it will release more, depending as the levels rise or fall in the bloodstream. So I have several different images of the pathways. I'm going to leave this one up here as we talk about it. And actually, I have one of my non-embedded videos. You don't have to say this. Oh, okay. until this last year, keep that from 7:30 to 12 and 12 to 5, 12:30 to 5. So this is really basic. You're supposed to be excited about this.
Woo!
This should say follicle and thyroid cell. There's a few errors in here. And I'm going to draw this out for you. This should say TSH, not TRH. So after we watch it once with the music, I'll play it again and turn the music off and talk. This is going really fast here. Okay. So now let's do that again. And I'm going to put on this here where I can pause it. Bring it to- So here is a simple cuboidal cell. Again, I'm going to draw this on the board, go over it with you. All right. This would be the basal membrane and a capillary. This is the lumen of that sphere up here at the top. Apical surface, and how do we increase apical surface area? What do we call those folds? Microvilla. All right, because they don't need to move, so they're not cilia. In the basal membrane is a pump, but it doesn't use ATP. And iodide molecules are taken up from the blood. Iodide is ions come in the food that you eat. So I grew up in the Philippines, and which was made up of thousands of islands. And individuals that lived along the coast of those islands did fine. But if they were in the mountain regions of Luzon or Mindanao and didn't have access to dried fish, they often developed hypothyroidism. Not because they didn't have the proteins to make the hormone itself, but they didn't have the iodide to take up. So in our salt, you'll see on many of the, unless it says natural sea salt, which is already iodinated, iodine has been added to it. So the iodine is taken up in a channel along with sodium. It's actually sodium that drives that, because the iodide is going against its gradient. And so it kind of gets dragged in there with the sodium ions. So let's move it along. So this is the iodide coming in. And now the cell. Remember, thyroid hormone is modified. Thyroid hormone is modified tyrosine. But what is actually made is a protein known as thyroglobulin. Pull this back here over. And so the cell takes amino acids and makes a long protein known as thyroglobulin, which has tyrosine on it, inserted into it, among the other amino acids. And at the membrane, as it's externalized into the lumen of that basketball-shaped follicle, the iodide is added to the tyrosine. All right, 1 or 2 atoms of iodide to each tyrosine. And then it moves out into the lumen of the follicle. And iodide is added, so that happens at the membrane. And there it's stored until TSH signals the simple cuboidal cell that the body needs more thyroid hormone. So this is, this molecule represents the thyroglobulin plus the iodinated tyrosine. And all we need is the iodinated tyrosine. So we have to liberate that from the thyroglobulin. The next step is fusion group? What do they break down?
Proteins.
Proteins, so they are proteases. Thyroglobulin is a protein. All right, so let's look at this now with that activity in mind. Let's slow it down and diagram it out on the board. So here is the simple cuboidal cell. Microvilli. This would be the lumen of the follicle. And all that stored thyroglobulin, iodinated tyrosine is called colloid. So that's what we call all that stuff that's in there. Along the base of the cell, and endocrine cells are always going to secrete out their base. That's where the connective tissue is. Remember, every epithelial cell is associated with loose, every epithelium is associated with loose, irregular connective tissue. So our blood vessels are in that loose, irregular connective tissue. Endocrine cells go out the bottom, not out the top. This cell's going to do both. But the final product will be out the bottom. Okay? So in this cell, coming from the digestive tract, we have iodide molecules. Iodide molecules are in higher concentration inside the cell than they are outside the cell. And yet, iodide moves into the cell. You may or may not have learned these terms in Bio 430, but we'll see them again when we talk about the kidney. Where is sodium in its higher concentration? Inside the cell or outside? Outside. So if we have a leak channel for sodium, or if we open a ligand-gated channel for sodium, it's going to go into the cell. So this channel is going to allow sodium ions to move in, two sodium ions. And by the way, it changes the shape and the charge characteristics of the inside of the channel. It will drag with it, up its gradient, an iodide molecule. So this is a sodium iodide pump that doesn't use ATP.
Is it because they're opposite attracted, so they're attracted to it?
That would be part of it. And so we have, this is where on our diagram in that video it said TRH stimulated. It should be TSH, it's the anterior pituitary that will work on this pump. And so since we have sodium and potassium pumps, we're called antiport, because they went in opposite directions, right? Sodium went out of the cell, and potassium went into the cell. So if that's an antiport, what would we call this one? A symport. And instead of active transport, we call it secondary active transport, or secondary transport. It doesn't require ATP for that to work that particular pump. All right, so on our diagram over here, this is where we are. And it's correctly labeled TSH sensitive. Okay. So now we've got the iodide, but we have to take our tyrosine, because thyroid hormone is iodinated tyrosine. Well, to support that tyrosine, which is an individual amino acid, it is inserted into a protein chain of amino acids known as thyroglobulin. So I'm going to diagram it over here. So we have a long chain of amino acids, and inserted bunch of other atoms sticking off on here. So this is thyroglobulin, and this is tyrosine. So that's the role. There's actually one of the diagrams that I have on here is a little bit more, shows the Golgi apparatus. So here's the iodide coming in. I used the other one, but it was closer to the video I just showed. Thyroglobulin is synthesized using messenger RNA into the ribosomal ER, modified in the Golgi, and packets of that come out here. So it's the tyrosine embedded in the thyroglobulin. Right? Right at the surface, we have an enzyme known as thyroid peroxidase. The thyroid peroxidase does two things. It adds iodide, ion form, iodide to the tyrosine, and it adds one tyrosine to another. So this is a new slide that I found last night. So pardon? So here's thyroglobulin with the tyrosine.
You turn the one light off?
Huh?
Thank you.
So the red line is the thyroglobulin, and thyroid peroxidase takes iodide and adds either 2 or 1 iodide to each tyrosine. So that's one thing it does. And the other thing it does is it will take one tyrosine, iodinated tyrosine, and add it to another one. Okay? So how many iodide molecule atoms do we have, ions do we have on that one?
4.
4. So that is T4, or thyroxine, tetraiodothyronine. I don't know how many other names we have for this. All right, so back to this image. So that's what thyroid peroxidase does. So out in the colloid, what we have is thyroglobulin with tyrosine and another tyrosine. And if we have that, that would be T3. And if we have that, that would be T4. So it may only add 1. It may only add 1 iodine, or it may add 2. So if 1 and 2 are added, we get T3. If 2 and 2 are added, we get T4. And you thought that using math would never help you out. Okay. So I think understanding it like this is a lot easier than figuring that out. But that's the 2 tyrosine molecules along the thyroglobulin. Yes?
So the red lines on the thyroglobulin are the thyroid peroxidase?
The red
Iodide.
gland are the
Iodide.
iodide.
Oh,
Okay, so that's now an iodinated tyrosine. But it's still associated with the thyroglobulin, which is the black one. So as a hormone, what we're going to see in the blood that is able to work is this. Or the one with 4.
T3?
T3 is the most active, T4 the most common. So we have to get from this to that. So it's stored in the colloid as the thyroglobulin with iodinated tyrosine, and then again as the body with low levels of thyroid hormone reaching the hypothalamus, and more TSH is produced. Not only does it stimulate the pump, not only does TSH stimulate the thyroglobulin, it causes the cell to transport free iodinated molecules back out. That requires a process known as endocytosis. Remember that from last semester? The cell membrane invaginates and then eventually internalizes. And this is still going to be attached to thyroglobulin. Okay, I'll put another one on there just for, I'm drawing circles now, I'm not drawing. And remember the lysosome in the video? So the lysosomal enzymes are going to come in, and when we break down proteins, what do we get? Individual amino acids, which is why tyrosine doesn't get broken down, because it's already a pretty small amino acid component. It doesn't have a substrate for that. So this starts to break, the lysosome starts to break down the thyroglobulin. And it releases free amino acids into the cytoplasm to be used again to make more thyroglobulin. And now we release the T3 or the T4. Into the blood. Pardon? Negative. What happens, and I skipped the step, I don't know why they have a positive there. What happens when it comes in is most of the iodide ions are actually bound together and have no charge. But as a single one, it's negative. Well, I don't know why they put a plus there. Yes?
Does the lysosome also break down the thyroglobulin, not just the protein?
No, yeah, that's what it's breaking down. It is breaking the thyroglobulin to individual amino acids, leaving the iodinated tyrosine molecules free. And then that is moved into the blood. So that's what's happening here. Now, even though it's an amino acid, it actually is lipid soluble. Not enough to cross the plasma membrane by itself, but still requires a transport protein, but enough hydrophobic that it's not soluble in the blood. So it has to be bound to a transport protein. All right, I'll get you in just a second. So in the blood, it will bind to several different kinds, but the most common one is known as thyroid binding protein, or TBP. Okay. So about 70% of the thyroid hormone in your blood is bound. About less than 1% is the most active form of T3. So if you say, I'm feeling really tired and sluggish and I'm gaining weight and I'm having problems with constipation and my hair is dull and not shiny anymore, and you tell your doctor you think you have thyroid problems, and he or she draws some blood and says, well, your hormone levels are fine, it depends on what they tested. You can test for just levels of TBP, the thyroid protein binding protein, or you can test the ratio of T3, T4. You may have lots of T4, but it's not as functional as T3. So if the doctor says, well, your T4 levels are fine, but you don't have enough T3, you might not know that. So there's all, the complication of dealing with the endocrine system. Yes? You had a question.
So what's actually happening when the thyroglobulin goes to doing it? Because the thyroglobulin already in the cell contains the tyrosine already in it.
Yes.
Before it leaves exercise.
It has,
right?
so this molecule right here is this one without the iodine.
Oh, okay, so it's picking up iodine from the liver.
That happens at the membrane with the thyroid toxins
Okay.
and the clumping of two tyrosine molecules together. Okay?
When did the PBD get attached to the nuclear?
Oh, that's in the blood, and it's probably, I would have to double check, but most of those type of transport proteins are made by the liver. So that would